Contrary to popular belief, cancers are not synonymous with tumors. To put it simply, all cancers are tumors, with the exception of blood cancers. However, not all tumors are cancers, for they are merely a type of tumor. Believe it or not, there are normal bodily functions that contribute to tumor growth. This includes the formation of blood vessels, also known as angiogenesis. You see, tumors release MMPs or matrix metalloproteinases that deteriorate the ECM or the extracellular matrix. This allows angiogenic factors and signals to reach receptors, which causes new blood vessels to form. The tumors then receive oxygen and nutrients through these blood vessels, allowing them to grow further.
With that information, we have decided to target the angiogenesis factors of tumors. We did this by investigating the anti-angiogenic potential of common water hyacinth, scientifically known then as Iconiac crassipes, and now called Pondaria crassipes. We figured that they are suitable for this experimental study after learning about the various anti-angiogenic phytochemicals compound present in the plants, including alkaloids, flavonoids, phenols, tannins, saponins, terpenoids, and anthraquinones. Another reason is that they are fairly common in the Philippines. Inhabiting its bodies of water, it's just that they are mistaken as water lilies.
We are student researchers from grade 12 Salca of the Strand, Science, Technology, Engineering, and Mathematics or STEM, and we would like to introduce AngiOver, an ethanolic extract out of water hyacinth leaves that has the potential to target angiogenesis, causing inhibition of tumor growth and formation. By being a cost-efficient inhibitor of angiogenesis, we are able to bring awareness to water hyacinths by giving them another purpose, which can solve the problem of them being evasive and causing bodies of water to overflow. Additionally, we also want to make use of more botanical plants here in the Philippines. But most importantly, our experimental study can contribute to the ongoing investigation of prevention, treatment, and cure for tumors and possibly even cancer. To proceed with the product making, we first bought and prepared all of the materials and equipment to be used. The list of the materials is as follows: water hyacinth leaves, scissors, paper towels, a baking tray, parchment paper, undenatured 96% ethanol, qualitative filter papers, beakers, cling wrap, and aluminum foil. Then, the equipment used were an oven, a blender, a rotary evaporator, and personal protective equipment or PPE, which includes medical gloves, medical masks, aprons, and hairnets.
Our closest and most convenient source of water hyacinths is the shore of Laguna de Bay. We gathered a generous amount of water hyacinth leaves that will be then thoroughly rinsed 3 times, twice with tap water and once with distilled water, to avoid contamination. Then, the leaves were dried with paper towels and cut into smaller pieces. These leaf bits were dehydrated in a preheated oven for about 20 minutes or until they turned crumbly. Take note that we wore our PPE or personal protective equipment throughout the entire process of product making and truly washed our hands regularly.
We put the dehydrated leaves in a blender to turn them into powder. An amount of 180 grams was taken from the product and added to 900 millimeters of undenatured 96% ethanol, keeping the standard ratio of 1:5. We prepared the concoction for maceration by keeping the ethanol from evaporating using cling wrap.
Macerations allows the anti-angiogenic phytochemical present in the leaves to be released into the ethanol that breaks down its cell wall. After this process, the concoction was filtered. For faster and more efficient filtration, we had multiple setups in which their filtrates were combined together to a total of 250 millimeters. After getting a schedule with Central Instrumentation Facility of De La Salle University, we bought the filtrate to them. They will be using a rotary evaporator to remove most of the ethanol solvent in the extract.
We also visited the Jose Vera Santos Memorial Herbarium of the University of the Philippines, Diliman's Institute of Biology, to get our plant samples identified and authenticated. The next day, we received an email from the institution with the certificate of identification for our plant attached to it. With this, we can officially confirm that the plants we use are indeed Pontederia crassipes. Our materials and equipment must be ready and complete before we buy the domestic duck eggs, since we had to start testing on them while they were still 12 days old. We went ahead and made a simple homemade incubator for the domestic duck eggs. Also, it wasn't that long ago when we finally availed an electrical digital microscope which will be used in observing and documenting the assays.
Before proceeding to the actual testing procedure, we had to make sure that our water hyacinth ethanolic leaf extract did not contain any more of the ethanol solvent, because the presence of ethanol in the extract will make it contribute to the blood vessel proliferation instead of inhibition. We tested this by lighting it on fire. The extract ended up being incombustible, which guarantees the absence of ethanol and means we can now start with the CAM assay.
The CAM assay procedure included the following materials: isopropyl alcohol, paper towels, tweezers, cutter, 1cc syringe with needles, medical tape, undenatured 96% ethanol, vitamin A capsules, water hyacinth ethanolic leaf extract, and 12-day-old domestic duck eggs. Equipment such as the homemade incubator and electrical digital microscope were also used.
Upon completing the equipment and materials, we went to Victoria Laguna to fetch a tray of 30 pieces of 10-day-old domestic duck eggs from Mr. Danton, who kindly candled the eggs to ensure their health and recommended we incubate them at 37.2 to 37.6 degrees Celsius. After transferring the eggs back to our location, we incubated them until they turned 12 days old. Once they were at the ideal age, we immediately started with the CAM assay by removing some of the albumin to make space for the treatments and avoid overflowing. The hole through which this was done was covered using medical tape to avoid leakage. Then, we carefully made a small window where the blood vessels were most visible and took a picture of them. Afterward, we applied the treatments to the respective setups and covered up the windows using medical tape before putting the eggs back in the incubator. The CAM assays were documented after 24 hours and 48 hours.
Once the pictures were complete and organized, they were all uploaded to ICOSA's CAM assay image analyzation tool to quantify the length and amount of branching points of the embryo's blood vessels. These are the outputs for R0, R24, and R48.
The control groups were able to show their expected results, both in terms of length and amount of branching points. Ethanol as the negative control did not inhibit blood vessel proliferation, while vitamin A as the positive control did. Surprisingly, the water hyacinth ethanolic leaf extract in different amounts was also able to reduce the growth of the blood vessels. We can also conclude that the amount of the extract is directly proportional to the reduction percentage of the blood vessels in terms of length and number of branching points, which means the more of the extract is added, the better the results are.